Assalamualaikum to all panelists. I am Intan, Aslinda, Danik. So today I would like to present my final year project, which the title is The Presence of Microplastics on Caulerpa Lentillifera species and Padina species. Background of study. Microplastic is any manufactured polymeric matrix or solid particles. The size range from 1 micrometer to 5 micrometer. So for Caulerpa Lentillifera species, it is edible algae and rich in polysaccharides. For Padina species, it is non-edible algae and it has potential to improve wound healing in vitro. Problem statement. Wastewater contains microplastic from various sources discharged into the natural environment. Caulerpa Lentillifera species and Padina species is a vector of microplastic into marine food webs. When it is consumed, microplastic will block the digestive tracts, diminish the urge to eat, and alter feeding behavior, all of which reduce growth and reproductive output. Significance of study to identify the presence of microplastic on algae, which is Caulerpa Lentillifera species and Padina species. Findings can be used as a benchmark of several microplastic populations in the sea. The objective first to determine the presence of microplastic on algae, and then to investigate the number of microplastic attached on algae, and lastly to identify the physical and chemical characteristics of microplastic presence on the algae. And lastly, scope of study focus on investigations of microplastic presence on Caulerpa Lentillifera species and Padina species which obtained from Dixon's Miller. Next, literature review. Study by Anderson et al. Stated that microplastic particles have recently been given attention as an emerging contaminant of concern internationally. Next, when other marine organisms consume other organisms which were contaminated by microplastic, the microplastic was transmitted and accumulated in the food web between organisms with different trophic levels. So next, the shapes and colors of microplastic found in cage culture and wild Caulerpa from Setu Wetland Malaysia are green thread-like, transparent thread-like, black thread-like, orange thread-like, white spherical, white fragment, and purple thread-like. From this study, black thread-like shape was the most abundant microplastic particles found in both cage culture and wild Caulerpa at different sampling locations, followed by transparent and blue thread-like shapes. Most of the microplastic found in noriform are fibrous, indicating that they may come from multiple sources including river inputs and sewage from coastal areas, and the decomposition of larger plastic fragments caused by local human activities. Lastly, Scope Sunny et al. Stated that visible light and IR imaging of polyamide fiber was identified, which the microplastic sample was taken from Lake Garda, Italy. The intensity of the following bands from the study are 3403 until 3225 NH stretch area, 2908 CH stretch, 1724 until 1596 C double bond O stretching, and lastly 1598 until 1528 N single bond H bending and C single bond N stretching. Next, for procedure A, 1 gram of seaweed sample was weighed and replication was done for 3 times. The sample was soaked for 2 hours in seawater, and then it was filtered using vacuum pump to filter any microplastic particles. Lastly, the filter paper was put in the petri dish for the analysis under the dissecting microscope. Procedure B, both samples was rinsed 4 times using deionized water. 10 grams of seaweed sample was weighed and replication was done for 3 times. The sample then was dried before the digestion process, and then 6 molar of NaOH were poured into the beakers containing dried seaweed. Both samples was put in the oven for 50 degrees Celsius for 2 hours and were placed in water bath, and it was shaken constantly during the incubation period. The sample was filtered using vacuum pump and fiberglass filter paper and was analyzed under dissecting microscope. Procedure C, 5 ml for each sample from procedure A and B were poured into the test tube, and then it was analyzed under the FDR/ATR for 80 scans per analysis. The sample were exposed to the radiations and matched with the variations, and then the sample will then be scanned using a computer to observe the result. For physical characterizations, Figure 4.3 shows the average of microplastic attached on algae surface of Caulerpa Lentillifera species and Padina species. The black trilla is the most abundant microplastic on Padina species with average 5.33 compared to on Caulerpa Lentillifera with average 3.0. The less abundant microplastic is red fragment, which is on Caulerpa Lentillifera species with average 0.23. There is no red and blue fragment microplastic was found on Padina species. So, compared to previous study from Sambe et al. 2018, results show that the average microplastic attached to the surface of vesicles is 3.99. Figure 4.7 show the average of microplastic in Caulerpa Lentillifera A and Caulerpa Lentillifera B. The highest average is black threadlight, which is 6.67 in Caulerpa Lentillifera B. The lowest average also from Caulerpa Lentillifera B, which are orange threadlight and pink threadlight. Both average is 0.33. From previous study on big samples collected from the high tide line, it contains significantly higher abundance of microplastic than at the low tide line. The microplastic in high tide line bead sediments with a mean of 1,323 was 4 times higher than those recorded from low tide line with mean 178. Figure 4.8 show the average mean of microplastic in Caulerpa Lentillifera species and Padina species. So the graph show the highest average is black threadlight in Padina species, and the lowest average was orange threadlight and pink threadlight, both with average 0.23 in Caulerpa Lentillifera species. The study aligned with previous study by Ibrahim et al., which most of the microplastic found had threadlight shape with distinctive colors which are blue, black, red, purple, green, orange, or colorless. Figure 4.9 show the transmittance spectra of microplastic on the surface of Caulerpa Lentillifera. At wave number 1030.88 cm-1, it is identified as polypropylene, and the stretching bands appear corresponding to C single bond C stretch and rock. This study can be compared to previous study by Zhang et al., which the PP was found at wavelength 1166 and 972 cm-1. This study can be compared to previous study by Zhang et al., which the polypropylene was found at wavelength 1166 and 972 cm-1. This study aligned with previous study by Magahes and Nunes et al., which previously were identified at wave number 2970 and 1912. At wave number 1506.85, it is identified as polystyrene. In this region between 1583, 1492 and 1451 cm-91, several stretching bands appear, corresponding to CC and aromatic rings. So lastly, figure 4.12 show the transmittance spectra of microplastic in Padina species. At wave number 1635.46, it is identified as nylon, and stretching bands appear corresponding to CO stretch. Compared to study by Zhang et al. 2019, nylon were identified at wavelength 1634 and 1538. So, as conclusions, the type of microplastic identified for Caulerpa Lentillifera species are polypropylene, nylon, and PVC, while for Padina species, the type of microplastic are nylon and polytetrafluoroethylene. For average number of microplastics on the surface of algae per 10 gram, Padina species has the highest average of black thread-like microplastic, with average 5.33 compared to Caulerpa Lentillifera species, which the highest average of black thread-like microplastic is 3.0. The lowest average number of microplastic on the surface of algae per 10 gram is red fragment, with average 0.23 and followed by blue fragment 0.67, which both is from microplastic on Caulerpa Lentillifera species. And then for average number of microplastic in algae per 10 gram, Padina species has the highest average value of microplastic, which black thread-like 8.23 average. So, the lowest average number of microplastic in algae per 10 gram is in Caulerpa Lentillifera species, which are orange jet lag and red fragment microplastic with both average 0.33.